Hello, everyone. A very good evening to all of you. And I welcome you all to yet another session of Brahmastra Series where we will be discussing about 2C. Okay. Can you please confirm whether I am audible to all of you or not? So, today we are going to discuss regarding genome organization on the first part. And once we have done with that, we will move on to 4B, that is cell signaling. So I hope so. I am audible to all of you and my screen is also visible to everyone. Correct? Okay, fine. So moving on to the first question from genome. Just one question I have added from cytoskeletal system which came in the last time exam. Just have a check on to this question whether you are able to answer or not. Now this is related to accessory proteins that came recently. So we have actin binding proteins that regulate microfilament turnover in a eukaryotic system. So you have to match the actin binding proteins along with their function. Try out. In my last session, I had discussed with you regarding the functions of different accessory proteins, whether it is coming from microtubules or from microfilaments. Of course, intermediate filaments we don't have as such, but in these two cases, yes, we are having a lot of accessory proteins. Can you please try on this question? So here's the options are given. So you can mark it as 1, 2, 3, 4. So you can give your answers as 1, 2, 3, or 4. Try out, everyone. So let's check it out whether you all are remembering the accessory proteins or not. Yes. Are you all able to answer? Good evening, Anusha. Good to see you here. So is there anyone who is trying out this part? This is the same part we had discussed in our last Brahmastra series. We had discussed regarding the accessory proteins. Good evening, Anjali. So you're not able to get it? So if is the image is clear, right? Coffeeline, trophyline, thymosin, and formin. So in the column 2, you are having binds ADP-G actin and catalyzes the exchange of ADP for ATP, binds preferentially to filament that contains ADP actin, assembles unbranched filaments, and binds to ATP-G actin, inhibiting the addition of actin subunit to the filaments. Are you able to get it? Now let me show you the options, the combinations. These are the combinations. Yeah, good evening, Reshma. So can I expect answers from your end or shall I give the answer? And to avoid any sort of confusion, yes, we have, in this question, we are having only the actin binding proteins. That means the accessory proteins present in actin. Anyone? I did not get a single answer from the for this question. Okay, very good. Anjali came up with the answer. Surinder, okay, fine. I got two answers and both of them as 4. Anyone else? Anjali, Reshma, would you like to give your inputs over here in this question? Okay, Anusha. Okay, let me give you the answer so that things has to be more clear. The correct answer over here is 3. That is, coffee belongs to what? This is your coffee, and it binds preferentially to the ADP binding portion, that is ADP active. Then we are having B to 1, that is profiling. If you remember, I told you in my last class also, profiling is related to Jeff, guanine exchange factor. But in this case, you are having the exchange taking place between ADP to ATP before going for forming, because first you have profiling comes that will do the exchange, and after that you are having the formine into which your trimers are getting synthesized. Trimer actin is getting synthesized. So like that you are getting a filament. So G actin will be the monomeric form and F will be the filament form. So like that it becomes a trimer. Then you are having thymosin, that is C to 4, which is responsible for binding to ATP G actin, thereby inhibiting the actin subunits to filaments. That means it causes disassembly, right? And the last one is formine. So formine indicates assembling in the unbranched filament. If you remember, I told you two proteins. One is formine, another is ARP2 bar 3. Both of them are involved with activation, but this will form unbranched filaments and this will form branched filaments. Okay, that is the difference. Otherwise, both of them, the working remains the same. The difference is that they will form unbranched to branched filament. Okay, so this was the only question which I wanted to ask you from cytoskeletal system. Let's move on to genome organization. Which one of the following statements about chromatin is not true? DNA winds approximately 1.65 times around the nucleosome. H2A, H2B binds to the entry and exit ends of the DNA in the nucleosome. Covalent modification of histones influences chromatin compaction, and non-histone proteins are part of mitotic chromosomes. Find it out. When we speak of questions related to genome organism, the questions are always direct. The only thing is that your basic fundamental has to be clear. So based on that only the questions are being asked. Okay. So if you know the organization, how the nucleosomes are formed, how histones are bound, what is the wrapping of DNA, how many base pairs are there. So these are some basic things you should be aware with respect to chromatin. So if you understand that, all the questions will be easy for you. Yes, find out. And another thing I want to point it out over here is, here they are asking you the incorrect statement. Sometimes what happens when we look into this term chromatin and look out the sentences, we forget that the question is being asked as not true. So always read the question thoroughly whenever you are attending any exam. Somewhere it is given, somewhere it might have asked the not true statement, somewhere they will ask you the true statement. So based on that you have to give the response. Yes, what is the answer for this question? I hope so the previous question was clear to all of you. Why the answer is 3. What should be the answer? Have you studied genome organization? Have you all revised this topic and come? because I can see very less responses coming up. Yes, Anupam, Reshma, Anjali, Anusha, Surender. Okay, Anchal. Okay, Anupam and Anchal has given the answer as 3. That says that covalent modification of histones influences chromatin compaction. Anjali has given non-histone proteins are part of mitotic chromosomes. Anyone else would like to give the answer over here? Would like to give your inputs? Yes, Surinder, Reshma. Okay, Sadia. 2 is incorrect, you are saying. Okay. Vanishri. Yes, 2. Okay. Let's check out the answer, which is the incorrect statement over here. Yes, and you are absolutely right. Whoever has given 2 as the answer. H2A and H2B binds to both entry and exit ends of DNA in the nucleosome. So, if this statement is incorrect, then can you tell me who or which histone proteins are responsible for binding to the entry and exit ends? You are able to imagine the organization, right? You are able to imagine how the nucleosomes are bound, how the DNA wrapping takes place. In that case, yes, in that case, what or which histone will be present in the entry and exit site? Can anyone tell me? So basically, the non-histone proteins, that is the regulatory proteins, definitely they are a part of mitotic chromosomes. Wrapping is also correct, 1.65 times around the nucleosomes. Then we are having covalent modification of histones influencing the compaction. Yes, depending upon the modification of histones, whether it is methylation, phosphorylation, or acetylation, it is going to affect your chromatin formation, right? Absolutely correct, Vanishri and Anjali. So, nucleosomes from all the eukaryotes, if you study, they are composed of 146 base pairs of DNA being wrapped. But the length of the linker DNA will be variable among the species because it can range from 15 to 55 base pairs. So, depending upon this pairs, we can say that together with histone H1 and the octameric histone will interact, giving rise to 168 base pairs of DNA. So, this makes the first statement correct. Then, linker histones. Why? This is H1, which is responsible for binding to the nucleosome at both the entry and exit sites of the DNA, which in turn is responsible for the formation of higher order structures. Okay, so I'll have a basic what is how exactly the organization takes place. You can look into this image. See, at the simplest level, chromatin is considered to be a double helical structure of DNA. So this is your double helical structure of DNA. Now, when this DNA is complexed with histones, it becomes your nucleosomes. So terminological differences you are getting. After that, each nucleosome will be composed of 8 histone proteins that will be wrapping the DNA around 1.65 times. Okay, so like this. Correct? And after that, you get a structure known as chromatosome. That means when your nucleosome is combined with the H1 histones. Okay, so when your histone H1, that is your linker histone, is combined, at that time you call it as a chromatosome. And after that, it will undergo compaction, giving rise to 30 nanometer fiber, 300 nanometer in length, where you will find loops are being formed like this. And after that, it will be more compressed and folded like this, 250 nanometer width. And finally, you will be finding the tight coiling of this 250 nanometer fiber. That is, you can visualize it under metaphase chromosome, metaphase stage of the cell cycle. So this is how your genome organization takes place. H1 will be there, Sadia. Okay. So if you look into this packaging, see, this is your double helical structure of DNA, right? Now, if you take this portion and visualize, you will find beads on a string form of chromatin, which is around 11 nanometer fiber. So this is considered to be first order of packaging. Next, what you are having 30 nanometer chromatin fiber, which is considered to be second order of packaging known as solenoid. And then you are having scaffold loops like this. 300 nanometer, and you are calling it as a scaffold loops. And if you are taking this portion and visualize, you can find condensed form is visualized. So you call it as a chromatid. And this chromatid is highly compressed in the metaphase stage of the cell cycle. So that becomes your chromosome. Okay. So remember this packaging and remember the sequence. Important to understand the organization of chromosomes. So next question in your screen. Which one of the following statements is not true about nucleosomal organization of core particles? The typical structure of DNA is altered in the middle of the core particle. Within the core particle, you'll find DNA being organized as a superhelix with 1.65 turns around the histone octamer. Within 30 nanometer fiber, you will find 6 nucleosomes per turn, which is organized into a two-start helix. And the last point, the N-terminal histone tails are strictly ordered and exit from the nucleosomes between the turns of DNA. Find out the correct answer. I mean, you have to find out the incorrect statement from this question. Find out. Yes, Sadhya, it is H1. So basically, you can expect questions from this nucleosomal organization. Then some part of it you may expect in the form of a cell cycle, like what is the situation of nucleus in different organisms, higher eukaryotes, lower eukaryotes, prokaryotes, like that. Earlier, they used to ask with respect to this heterochromatin and euchromatin, but nowadays they are not asking because those have become very familiar to all. So they have stopped asking. But this organization part, every time a 4 marks is expected. Yes. Can anyone give answer to this question? Do you know what is a two-start helixes? Anyone? Anyone aware of two-start helix? Okay, I can see Anupam has given the answer. Can anyone tell me what is the two-start helix? Whoever has given the answer so far, can anyone tell me what is a two-start helix? Confused between 1 and 4. Okay, try and get it one answer. Don't know. Vanishri, are you aware of this two-start helix? Rubin, 4 is giving the answer. Okay. Anjali, Anchal, today it seems questions are tough or you are not able to answer. I'm not sure. Usually I used to get very good responses from you all, but today response is limited. Okay, let's check out the answer. The correct answer over here is the N-terminal histone tails in a core particle are strictly ordered and exit from the nucleosome between the turns of DNA. So let's find it out what is the organization is. The N-terminal histone tails are highly flexible and unstructured, so extending outward from the nucleosome core. And these tails are exposed to outside because they are responsible for undergoing epigenetic modifications. Do you know what is an epigenetic modification, sir? Yes, definitely. Literally speaking, these are not tough questions because the question which I had asked you was a 2 marks part B question. Direct question it is. The only thing is that maybe somewhere you are lagging with the basic fundamentals of the organization part. So just revise that portion. You'll get the answers. Okay. Yes, you are correct, Vanishri. Yeah, what is this epigenetic modification is all about? Yes, you have to revise this portion mandatorily because without understanding this portion, it becomes a base to understand unit 3, your molecular biology, which is full of chromosomes, where you have to understand replication, transcription, translation. So this is the base onto which your unit 3 is constructed. So you have to be very clear with this topic. Modifications is based on environmental changes. Okay. Okay, let's have a look onto that. So there are various epigenetic modifications and interactions with other proteins that regulate the structure of chromatin as well as gene expression. Okay, fine. See, in assembling a nucleosome, we have got histone folds present. So if you look into the image, this is the arrangement. Okay, these portions which you see, these portions and all, these are nothing but a fold. What is the necessity of a histone fold? So that they will help in accumulating the histones together. See, if you look into this image, right? So initially your H3H4 tetramer will come, then dimer H2H2B dimer will come. So together what you are going to get? You are going to get an octamer. So you can see, and everywhere the N terminal is given outside. Why this N terminal is given outside? Any specific reason? Because in this protein, it is in your N terminal which undergoes modifications. Modifications in the sense we can say methylation, phosphorylation, acetylation, right? So it is in this portion this modifications takes place, and that's the reason they are exposed to outside. Okay, so coming to this point, the histone fold first bind to each other forming H3H4 and H2A H2B dimers. Now this H3H4 dimers combine together forming a tetramer. Now this tetramer will combine with the dimers forming an octamer around which the DNA is bound. Now how this DNA to histone interaction takes place? By means of hydrogen bonding. And hydrogen bonding takes place between whom? Between the DNA along with the histone core present in each nucleosome. Okay. Then nearly half of these bonds form between the amino acid backbone of histone. So which amino acids you will find? here it is mentioned that the hydrogen bonding takes place between the amino acid backbone of histones to that of sugar phosphate backbone of DNA. So, which amino acid are they referring to? Yes, you are correct, Amit. Shrinkala, Anupam, Anchal, Satya. Yes, you are correct. So, what is the amino acids? I want to know histones are made up of what kind of amino acids? Very simple it is. Everyone can give the answer. Which amino acid you are having? We know that DNA is negatively charged. Why? Because of this phosphoryl group. So obviously, in the universe, opposite attracts. That means if you have a negative charge over here, you should have a positive charge in the opposite. So what kind of positive charge or positive amino acid you will get it over here? Yeah, histidine it will not come. But you have other two amino acids, that is lysine and arginine. Right. So apart from hydrogen bonding, this DNA to histone interaction can also take place via hydrophobic interactions as well as salt linkages. So remember three major what we can say bonding that contributes to the stability of interaction between DNA and histone. That is, one is hydrogen bonding, second is hydrophobic interaction, and third is salt linkages. Okay. So this is the arrangement. So first your tetramer will come and join, and below to that dimers will join. So N terminals will be exposed outside so that the modification can take in place. Because of the side chain, the interaction of the side chain, Amit. Then I had asked you about the two-start helix. So, two different models were proposed in order to understand 30 nanometer fiber. One was the solenoid model and the other one is the helical model. So this helical model is known as the two-start helix. Now what does it say? In solenoid model, a linear array of nucleosomes are coiled, as you can see. The arrangement, linear arrangement you can see. But this helical model or the two-start helical model, as the name indicates, it looks like a helix. So your nucleosomes are arranged in the zigzag pattern, forming supercoils and twist. Okay, so this is your two-start helix. Yes, Anupam, you are correct. That is because of the bulk ring. Histidine is having the bulk side chain, right? Yes, that is also one of the reason for that. Okay, next what we are having. So you are able to understand, right? The organization so far, whatever I had told you, the organization of genome, this helical models, the packaging, how the nucleosomes are arranged in the DNA. You are able to follow, right? Everyone able to follow, right? Okay, fine. Very good. So next what we are having? Okay, this is just for an information. Yes, we are going to have an exclusive cloning vector and recombinant DNA hands-on training taking place on 9th of September at 10 a.m. in Protein Design Research Lab at Bangalore. So, according to your convenience, you can attend either online or offline. Similarly, since it is being said that yes, we are having exams in the month of December, so we are launching a Raftar batch where it will be covered with only the important topics from examination point of view, which is starting from 11th of September. So all the important topics from all the units will be covered under this session. So if you want to know more about it, please dial on to our toll-free number and get more information regarding the same. Okay. So moving on to our next question. Okay. This is also based upon the nucleosome only. See, basically the questions will come based on that only. Nucleosome is the fundamental subunit of chromatid in eukaryotes. And these are some statements based on that. I think so far, whatever I explained to you, if you have understood it clearly, maybe you will be able to give the answer here. What it is? Generally, a typical nucleosome contains approximately 200 base pairs of DNA and two copies of each of these histones. 146 base pair of DNA per nucleosome is strictly maintained across the organisms. Histone octamers are not conserved during or after the replication. However, H3 to H4 to tetramers are. Variants can be identified in all the histone proteins, that means all the core histones excepting your histone 3. While wrapping around the core histones, the structure of DNA will be altered in the middle of the nucleosome and exhibits increase in the number of base pairs per turn. So what you need to identify over here? Okay, fine, Anupam. So what you need to identify over here? You have to find the combination of statements. Which combination of statements are correct? ACE, ABD, BDE, and ACD. So like that it is given. You just first look on to the statements. I think you can guess out yourself which statements are correct. Find out. Okay, I can see Anchal has given the answer as 1. 1 means a combination of AC and E. Okay. Others, try on. Try on this question. Very interesting question. I think many of you will be able to correlate it with what I discussed previously. The basic nucleosome organization. These kind of questions you can expect in the exam. This is a 4 marks question. Came in the exam. Okay. And sometimes they will combine this part with unit 3 molecular biology also. I have just added one question as a sample over there related to gene expression and chromatin at the end. So try on this one, everyone. Yes, Anupam, Reshma, Anjali, Vanishri, Anusha, Surender, Amit. Just find out the statements which you feel correct. You can go for that. Okay, I can see two answers coming as two. That is A, B, and D. Okay. So two answers, two different types of answers. One I got ACE and one is ABD. Okay, I can see Anupam has given the answer. Anjali, okay, majority of you are saying as two. That is A, B, and D are correct. Have you studied about histone variants? I think that you might have studied under molecular biology. Histone variants. Because there you have to study about the remodeling and all, chromatin remodeling and all, that is correlated with gene expressions. You might have studied over there. Okay, let's check it out. Okay, let's check it out, the correct combination over here, because I can see some confusions coming over here. A, B are correct. Okay. D is not true. Okay. So let's find out. So the correct answer over here, as mentioned in the very beginning by Anchal, yes, you are correct, A, C, and E. So a typical nucleosome contains approximately 200 base pairs of DNA. Why it is called as approximately 200 base pairs of DNA? Because if you are connecting it with linker histones, as I told you, the numbers will be varying. So they have just given an approximate figure. 146 is present per nucleosome is strictly maintained across the organism. So is it possible if A and B both can be correct? 146 base pairs per nucleosome is strictly maintained across the organisms. When you read the sentence from here itself, you can get that one of them has to be correct. Only one of them. Then, histone octamers are not conserved during or after the replication, but H32 and H42 tetramers are. Variants have been identified for all the histones excepting for H3. Then E, while wrapping around the histones, the structure of DNA will be altered at the middle. So definitely there will be increase in the number of base pairs for turn. Yes, let's look into the explanation now. So as I told you, A, C, E are correct. So why statement B and D are incorrect? See, the length of the DNA wrapped around a nucleosome core particles is relatively conserved, but it is not strictly maintained because in some cases the linker DNA could be shorter or longer. And additionally, if we will go for the histone variants like H2A, Z and all, that could cause variations in the length of DNA. So that's the reason, even though length of the DNA per nucleosome core particles can be said as conserved, but again, it depends upon the genomic and epigenetic context. That means based upon the organism, based upon the number of linker DNA, number of not the base pairs present in the linker DNA, and second, the presence of histone variants. And why statement D is incorrect? Because we are having variants of histone 3 available in eukaryotes in the form of 3.1, 3.2, 3.3, SEN-PA, and H3X. Okay, so that's the reason we have statements A, C, and E as correct, rather B and D as incorrect. And the reason why it is incorrect, here it is provided to you. Okay, fine. So let's move on to another question. Chromosome organization becomes clearer from a series of biochemical, electron microscopic, and X-ray crystallographic studies. So when interface chromatin is isolated in slow salt buffer and observed under electron microscope, 11 nanometer beads on string organization is C. Beads on string organization. Interface chromatin directly observed under electron microscope was shown to be 30 nanometer fiber. So when these histones are depleted from metaphase chromosome and visualized, it shows a huge number of loops present associated with scaffold. So based on this observation, following interpretations were made. 11 nanometer fiber is formed when nucleosomes are brought closer by scaffold. 30 nanometer is formed by zigzag organization of nucleosomes of 11 nanometer, or 30 nanometer makes a solenoid packaging to form metaphase chromosomes, or it is organized into loops due to SARS. What is SARS? Are you able to understand, or do you remember the SARS? What is SARS? SARS getting associated with scaffold proteins and coming closer. What is SARS? Can anyone can answer? SARS scaffold, no, scaffold associated region. Where it is present? It is present in your chromatin, where these scaffold proteins will go and bind. So they will have a specific location where it will be bound. Okay, so you have to identify the correct combination of interpretation here. Find out. Identify the correct combination of interpretations over here. So initially I had discussed with you the packaging and all, right? So if you remember that, apply that concept here. Yes, matrix associated region also we can say, that is we call it as Mars. Both are same. So what is the correct combination over here? Just try to remember the packaging part. You'll be able to get it. Not able to get any one of you? Try on. Okay, Reshma came up with an answer. See, don't be afraid whether it is right or wrong. You just say, at least you should have some information regarding the same, right? And since the beginning of this session, for the past 40 minutes, I'm discussing about genome organization only. So, definitely something might be there. Something might be there for you to reconnect it with this question. Okay, fine. Okay, I can see 3, 4, 2. 4, again 4. The main reason why you are not able to answer it, the question, you have to be very clear with the basic fundamentals. That is very important when we speak of chromosome organization. Okay, still can see many of you are giving the answers. See, even though right or wrong, right, you will be confident. Wrong, you will learn how to interpret an answer, or you will not make the same mistake again when you come across any relevant questions like this. So that's why speak off, give the answers. No one is going to say you anything. Okay, this much only. Let's find out. The answer is 4. As I can see, many of them has given the answer, that is 2 and 4. 11 nanometer interface chromatin is formed by zigzag organization of nucleosomes of 11 nanometer fiber. That's this point. This is what I told you in the first order, second order, third order packaging. Initially, you will be having the DNA beads on a string, that is 11 nanometer. Then you are having 30 nanometer. Then you are having scaffolds. Then you are having condensed form. Then highly compacted structure. So from that, you can easily get the answer 2. Next, 30 nanometer fibers can be organized into loops. The next organization. That's what I told you, due to SARS getting associated with scaffold proteins and coming closer. So obviously, if statement 2 is correct, 3 will be incorrect. You can call it as a solenoid packaging, but they have mentioned as metaphase chromosome. Metaphase chromosome is the last stage. This is the second order of packaging. So that becomes incorrect. And scaffold has got no role in the initial stage. It comes only after this 30 nanometer fiber. Okay. See, statement A, 11 nanometer fiber is a well defined structure and is thought to arise from interactions between neighboring nucleosomes, that is, via electrostatic interaction. Scaffolding proteins will be playing a role, but in the later stages in the organization of chromatin, that is, in 30 nanometer or after 30 nanometer stage. Statement B. The 30 nanometer fiber is thought to form when this fiber, 10 or 11 nanometer fiber, will undergo further compaction, resulting in the nucleosome adapting to a zigzag organization. Now, this is relevant to what? To start helical model or the zigzag model. And this is stabilized by histone to histone interactions, and the resulting fiber has a diameter of about 30 nanometer. Third statement, the organization of chromatin into 30 nanometer fiber will be a one step in the process of compaction. But the structure of metaphase chromosomes is even more complex. That's what I said to you. It comes in the last stage, not in the 30 nanometer stage. And finally, the statement D. This 30 nanometer fiber is thought to undergo further compaction and folding during the interface stage. And at that time, what I told you, during that stage, you will find loops like this. So that's the reason statement D is also correct. This question is clear to all of you. Why statements 2 and 4 are correct and why statements 1 and 3 incorrect? I hope so you all got it. Whoever has given the incorrect or could not give the answer. Got it, everyone? Yes? Yes, Reshma. Okay, fine. Very good. Then the last question, I think, from Genome Organization. So the following statements are made with the reference to the structure of nucleosome. So here, statements again. The histone tetramer in the core of nucleosome composed of these H2A, 2B, 3 and 4. N-terminal tails are believed to stabilize 30 nanometer fiber. Post-translational modification of N-terminal tails as well as globular domains of the histone modulate transcriptional events. Zigzag model, the 30 nanometer fiber, the linker DNA circles around the central axis of the fiber as the DNA moves from one nucleosome to that of the next. So what should be the correct combination of statements over here? If you see the pattern, the questions that are being asked from Genome Organization, whatever questions I had asked you, all the questions are somewhere correlated with each other. So when you know that connecting link regarding this organization, you will not find it difficult anymore. That's the reason repeatedly I am saying revise the topics. Revision is very important because today you have completed with unit 2, switching over to unit 4 means you should be in touch with unit 2 in order to avoid forgetting the topic. So for your convenient, if you want, yes, these are the options provided to you. B and C, ABC, C and D, B and D. Okay, easy to remember this option. B and C, ABC, C and D, B and D. Find out. Try out because so far we were discussing about the same only, right? So I don't think so you will face any difficulty. All you need to find is the connecting link. As I always say in the class also, try to establish a connecting link among the topics. Then only you will be more familiar with the topics over here. Okay, I can see Reshma has given the answer as 2. What is 2? ABC. Okay. Read the statements properly. Maybe you'll be able to get the answer because I have discussed about the histones. I have discussed about 30 nanometer fiber. I have discussed about the structure of this histone proteins. And I have also discussed with the zigzag model and solenoid model. So what has to be the correct answer? Okay, I can see Vanishri, Suresh and Anupam gave us answer 3. What does 3 says? C and D only. Okay. Any other answers from your end? Any other answers from your end? Okay, let me give you the answer. The correct answer over here is 1. That is B and C only. Now, first thing I want to make you realize it. This is known as tetramer, or is it known as octamer? What is the correct answer? We call the histone core as a tetramer or as an octamer. It should be octamer. So, obviously, the statement is incorrect. According to the zigzag model, DNA circle, the linker DNA circles around the central axis. Circles around the central axis means what? Somewhere we can correlate it with this helical formation of the fiber as the DNA moves from one nucleosome to the next. Not, we cannot say exactly as helix, but yes, like this it will be there. Like this it will be there. So this becomes which model? This becomes your solenoid model, which I had shown you earlier. So obviously this statement is incorrect. And obviously the post-translational modifications of the N-terminal tail is responsible for undergoing lot of changes. Then N-terminal tails are believed to stabilize the 30 nanometer fiber by interacting with adjacent nucleosomes as well. That is also possible, right? So that's the reason B and C are correct statements. So when you read the question, read the statements carefully. Clearly tetramer is written, but we know that it's an octamer. So this is, you can eliminate easily. So histones H2A, H2B, H3 and H4 are known as core histones. And core histones exist as dimers, and 4 dimers come together forming an octameric nucleosomes. Linker DNA circles around the central axis as the DNA moves from one nucleosome to the next. We call it as a solenoid model, not the two-start helix. Okay, that's you understood, right? So like this, you have to approach a question, understand a question, and give the answer. Okay, I hope so. You all are following this organization part. See, the majority of modifications occurs in the N-terminal region. So this is your histone full domain that I told you is necessary for combining the histones together when they are wrapping around the DNA. And these are the different modifications taking place. So there are occasional modifications which can take place in the histone fold. Usually histone fold domain is for attachment. Very rarely only the modification takes place over here. One such exceptional case you can see H3, where the methylation is taking place at the lysine 79th residue. In this case also, lysine 59th residue. So that is a rare case when the where the modifications takes place within the fold domain. Usually it takes place in the N-terminal region only, as you can see. Right? With comparison. Next. Yes, this was the question I was saying about chromatin remodeling. It alters or slides the nucleosomes but cannot displace it. It's an energy driven, developmentally regulated active process. Acetylation is a reversible process in which each direction of reaction is catalyzed by different enzymes. And in general, acetylation reduces their affinity for DNA, destabilizes the chromatin structure, causing transcriptional repression. And lastly, phosphorylation of serine 1 of histone H2A has been associated with transcription repression. So, this is a topic where you can combine unit 3 and 2 together. Why? Because they are speaking about gene expression. So, gene expression is mainly taught under unit 3. Chromatin remodeling, that is also comes over there. So, but it is having a connecting link to the structure of chromosomes, and that's the reason I shared this question with you. Are you all aware of chromatin remodeling? First, tell me that. Are you all aware of chromatin remodeling complex or the significance of chromatin remodeling complex? Are you aware of this term? Oh, you are asking for the same slide. You want the options. Okay, okay. Next time onwards, Vanishi, I will have a check on that. Okay. The thing is that questions are a bit larger, and if I will shrink it, maybe it won't be visualizing or not. I'm not sure. That's why I split into 2 pages. But yes, next time onwards, I'll have a check on that. Okay. Options. These are the options: ABC, ACD, BCE, and CDE. Okay. ABC, ACD, BCE, and CDE. Okay, let me give you the answer. The correct answer over here is 3, that is B, C, and E. So, first of all, what is chromatin remodeling? Chromatin remodeling is a system that is responsible for regulating your gene expression. That means providing access to your transcription factors to carry out gene expression. So that is again depending upon the requirement. See, chromatin remodeling is the dynamic modification of chromatin structure where they are providing access to transcription machinery proteins. So thereby controlling the gene expression. So how this remodeling can be taken place? Simply by undergoing histone modifications by specific enzymes like we are having HATs, deacetylases, methyl transferases, and kinases. Second, ATP dependent modeling can also be carried out simply by moving the nucleosomes or removing the nucleosomes or reordering the nucleosomes. And the last point, histone acetylation. We know that acetylation is responsible for neutralization of the charge. So, whatever the positively charged amino acids are present in the histones, they will be neutralized. So, if they are neutralized, the interaction between DNA and histones will be reduced. So, what happens? Your DNA becomes loosely packed. So, when this loosely packed DNA you will find or chromosome you will find, they will be having access to the transcriptional factors. So it results in gene expression. Got it? I hope so you all got it. Okay, then I thought to start with unit 4 also, but it's already going to be 6 now. So I will give you the questions in the next class. Okay? In the next class, we will have a discussion on this part, on unit 4. So far what I felt is genome organization needs to be revised by all of you. Badly required. Please go through the PPTs or recorded lectures or these YouTube series and try to understand the concept in a better way, because as I told you, this is a very important topic, not only from examination point of view, but also to understand other units as well, as this becomes a foundation for that. So make sure that you study properly with genome organization. So this was enough of questions I had asked, and no more questions will be coming from Genome Organization. In case I found, I will be definitely sharing with you. In the next session, I want you all to study cell signaling and come, because I'll be asking questions from cell signaling, all the signaling pathways. Okay. Okay, then see you again in the next session. Till then, revise, revise, and revise. And please work out MCQs on a regular basis. Okay, thank you so much, everyone.